So translation can only begin if mRNA is exported out of the nucleus. In order for such event to happen, mRNA needs to be processed. We discussed the addition of a 5' GTP cap and also a polyadenylation of the tail. And you can see various proteins are going to come together and they are going to translocate that mRNA all the way through the nuclear pore and then release the molecule in the cytoplasm for translation. We have three main components that are going to be carrying out the translation process. So we have the actual mRNA, and we have tRNA, transfer RNA, that's going to deliver amino acid to the synthesis site, and then we have ribosomes. So we're going to discuss all these components one at a time. So first, let's go back to mRNA. If you notice, the actual structure of mRNA is as follows. So we have a 5' GTP cap followed by 5' UTR, which is that untranslated region. This is where ribosomes are going to bind to mRNA and initiate translation. So we know that this is the beginning of the message. And then the message comes in the form of codons. So we have a triplet code here. So these three letters, ribonucleotides, are going to make up one codon. So this is a codon. One here, here's another one, here's another one. So we would have to read the codons to figure out what amino acid they code. And we're going to examine how we do that in a little bit. And then notice at the end of protein coding sequences, we have another sequence that's what we call 3' UTR, untranslated region, and then followed by poly A tail, which we already discussed in our previous lecture. So charged tRNA means we have an amino acid attached to the 3' end of this molecule, and the purpose of transfer RNA is exactly that, to deliver the amino acid to the synthesis site. So now, in order to decode the messenger RNA, we need to be able to understand the genetic code. So this genetic code applies to all forms of life, and it is the language of mRNA that's encoded by 4 different types of ribonucleotides. It's pretty amazing when you think about it. It's only 4 different types, and it applies to all the living things on this planet. So we have A, C, G, and U. And notice we're going to get 20 amino acids as a result. So we have a total of 64 codons and only 20 amino acids. So it means the code is a bit redundant. As you can see, sometimes we can change that third letter in a codon and still get exactly the same amino acid. So when that happens, we say silent mutations took place. So that way it doesn't really affect the protein. So again, we have 64 codons, and 61 of them will provide us those 20 amino acids. And if you can see here, we have 3 codons that are going to signify the stop of translation. Okay, so how do we actually decode the codon? How do we figure out which amino acid it codes for? So we can use a wheel or a table. And here's an example. So let's say we have a codon that starts with A, C, and U. So now remember, as I said before, make sure that you read the codon in the direction of 5 to 3. So I'm going to assume this is my 5' end and this is going to be my 3' end. So it means we're going to have to read it this way. ACU. So we start in the center, that's the inner circle, and then we go outwards. C, and then the third letter will come from here. So it means the amino acid that ACU codes for is threonine. And then to find it in a table, we choose the first letter from the column. So here's my first letter here. And then we're looking for C. Pick the letter from this column here. So this is the C. And then make these guys meet here in the box. And then, of course, the third one you can choose from this column here, but you can easily see that this is your ACU and it codes for threonine. So now, if you were given a codon, and notice the prime ends appear to be like this. For example, this is going to be your 3' end and this is 5. Remember, you cannot read it the way we just did. You must go this way. So you gotta read UCA. So it would be UCA. So that means it codes for serine. So it's a huge deal to make sure that you read the messenger in the correct direction, because you want to act like a ribosome. That's how they actually read the messenger RNA. Okay, so the tRNA is another component that is going to help mediate translation process. So tRNA has 4 key regions, and we're going to be concerned mainly about those 2. The acceptor stem that carries the actual amino acid at the 3' end, and then the anticodon loop. So this is the loop that contains anticodon. So anticodon will be complementary to codon. So codon is found on mRNA, and you can see here, yes, we have those prime ends. So there's my anticodon GAA, so it means this end means 5' because the end of the molecule is right here. Imagine if you stretch out the entire molecule, it would be like a string. So this is your 5' end, and the A would be sitting at the 3' end. So it means the corresponding codon would have CUU, because it's complementary base pairing rules here, G with C, A with U. And then you can see this end is going to be 3' and this is 5', because you have to match it. It's all complementary. So in order to determine the amino acid that this mRNA codes for, and therefore this tRNA is going to have to deliver to the synthesis site, remember, we must read that mRNA from 5 to 3 directions. So we basically would be reading it like this, UUC. If we look it up in a table, yes, indeed, we're going to find that it codes for phenylalanine. And then we have the other 2 loops. And if you're curious what their functions are, you can pause here and go ahead and read about it. So there are a couple of other things that I want to mention about the tRNA is the fact that it has the anticodon, and the anticodon is complementary to codon. So when the matching occurs during translation, that matching does not have to be perfect in one way. Third letter can actually vary. So let me give you an example. If you have anticodon that has AGG, a perfect match in the codon would be UCC. That would be a perfect match. And however, we can use the same tRNA even if your codon would read UCU, because C's and U's are single ring type of bases. And the G is the actual double ring base. Remember, we call that purine, and the C's and G's would be pyrimidines. So it means we have some room here for flexibility. We don't have to have that perfect match. And this, how does this help in translation? This eliminates the need for us to have all 64 different types of tRNAs to carry out translation. Because remember, we have 64 codons, so we would technically have to have 64 tRNAs. But because of this imperfect match and wobble position, we can actually use fewer than 64 tRNAs. So now the question is, how do we actually attach amino acids to tRNA? How do we charge them, or how do we load them? Well, there is an enzyme in the cytoplasm, and this enzyme is called amino acyl tRNA synthetase. So this amino acyl tRNA synthetase will have 2 active sites, one for the actual amino acid, so it's going to have a very specific shape to be able to recognize that. And then there is also another very specific shape to be able to fit in the tRNA. So, and the way tRNA is going to communicate with this protein, with this enzyme, is by forming hydrogen bonds between anticodon and the R groups of the protein. So that's how they're going to know this is the actual match, and this is the tRNA that's supposed to be sitting in the site to be able to attach the amino acid to it. So without going into too much detail, notice the ATP is going to be hydrolyzed and used for energy, and then here's your formation of that high energy bond between the tRNA and the amino acid. And we say now we have a loaded or charged tRNA that's ready to deliver that amino acid when the codon dictates in the mRNA. So keep in mind that specificity between the tRNA and its amino acid is extremely important, because if the rules weren't followed, we obviously would have errors in the amino acid chain. So now the ribosomes are catalytic machines that are going to be responsible for synthesizing the protein, one amino acid at a time. So they come in pairs. We have two subunits. So we have a small one and we have a large one. Small subunit is going to bind to mRNA and also provides that framework on which the tRNA can be matched to the codons. And then we have the large subunit, which will come, which has these three important sites: A site, P site, and E site. And you can see think of A site as if it's an amino acid site, because that's where the incoming tRNA is going to sit in. And then the P site, think of it as if it's polypeptide or formation of peptide bond site, because that's exactly what's happening there. And then the E site is for exit. So we're going to go through the steps of translation, but also before I go and talk about it, I want to point out that ribosomes are not organelles. They don't have their own membrane. They are basically proteins, polypeptides, lots of them, up to 50 of them can be in there. They are highly folded into this globular structure, and then they also have rRNA on the inside. So the rRNA can actually talk to mRNA during translation. So translation involves three steps. We're going to say initiation, elongation, and followed by termination. The initiation of translation process happens when small ribosomal subunit is going to bind to the mRNA. And it's always at the 5' end, because remember we had that 5' GTP cap here going on. We also have the 5' UTR region. So this is that region where a small ribosomal subunit is going to bind and then initiate translation. So the very first tRNA that arrives to the scene is going to be tRNA that carries methionine. So this is the amino acid, and this is basically a start amino acid specified by a start codon. So what is that codon? AUG. You can see it right here. It's kind of small, but here it is. If you look in a codon chart, you're going to see that AUG codes for methionine. And we already said that amino acid tRNA synthetase puts these guys together in a cytoplasm and they're ready to go. So this would be your initiation complex. And then what we have, the large subunit is recruited to the scene, and both these units are going to sort of join together like a puzzle piece. No covalent bonding happening there, but just like a puzzle piece that's going to hold everything together and read that mRNA. So the next process that happens after the initiation, we have elongation. So it means we can read the next codon. So the next codon is CCG. I know it's really tiny. Hope you can see it. CCG, and if you look it up in the codon chart, you're going to see that CCG codes for proline. And the anticodon that we need, that's going to be within that tRNA, that's going to deliver the proline, it's going to be GGC. So as you can see, this is a perfect match. So now the incoming tRNA is going to enter the A site, and now the magic happens. When I say the magic, there's going to be a couple of reactions that are going to be happening. So there's a reaction that removes methionine from tRNA that came in. So this bond is broken. And then there will be a formation of peptide bond between the amino acids. Remember, it's almost like we're linking these beads on a string, and the way we hold them together is by forming a peptide bond. So this is where that reaction happens, meaning at the P site of the large ribosomal subunit. And then once we have the dipeptide formed, notice the empty tRNA will now go into the exit site and eventually leave the ribosomal subunit. And now this entire complex will be able to read the next codon. So this would be UAU. You would have to look it up in a table and see what it codes for. And another tRNA would come in bringing a proper amino acid. And we would say there is a linkage between the amino acids, so it means we are elongating a polypeptide chain, one amino acid at a time, as we proceed and read that messenger by reading those codons. Okay, and eventually this ribosomal subunit, both of these units, are going to run into a stop codon. So stop codon means the end of translation. Release factor is going to bind to the A site and cause different reactions to take place, and one of them is going to be the release of the polypeptide chain. And then all of the components are going to dissociate. So now I want to remind you that ribosomes are sitting on the outside of ER. We call that rough endoplasmic reticulum. And they are typically feeding that growing polypeptide chain into the cavity of the ER. And this is some modification of the protein can take place, such as glycosylation, meaning addition of sugar chains. And then you can see the protein is folded and packaged into a little vesicle, and that vesicle is pinched off and most likely is going to be traveling to Golgi for further modification and sorting, and then final destination. It could be secreted out of the cell, or it could remain within the cell depending on the protein that was produced. I want to go back to the structure of amino acid. This would be a great way, a good time to review our biochemistry unit. And if you guys remember what amino acid looks like, here it is. So we have a dipeptide here, and now we're trying to add another amino acid to the chain. And notice that each amino acid will contain this alpha carbon, and then it has amino group on one end of it, and then carboxyl group on the other. And this is the R group that's going to be unique to each amino acid. So let me do one more. So here's the alpha, amino group, carboxyl group, and here's your R group. Alpha, and you can see R group. So these R groups are going to be interacting to form a tertiary structure, that 3D shape in the protein that's going to be a functioning type of form. The way we link up two amino acids, if you recall, carboxyl group is going to be reacting with amino group, and this is where the water is going to be forming and released. So this is really a condensation reaction, and we say this is where the actual peptide bond is going to form. And if you remember now, going back into and looking at the P site of the large ribosomal subunit, you're going to see that this is what that reaction is taking place. Okay, so let me see if I can pull this up really quickly and let you visualize what I have just discussed as far as elongation. So here we have the complex that has already been formed. First amino acid delivered to the scene, which is methionine because of AUG codon. Large ribosomal subunit binds, forms a large complex that's ready to do translation, and we have all the sites available. APA, here comes the tRNA, brings a proper amino acid based on a codon, and then the bond between tRNA is going to be broken, release that methionine, and transfer or form a bond with proline. So this is that peptide bond formation that is happening. And then the entire ribosomal subunit is going to glide over and you read the next codon, which is UAU, and you can see the other tRNA just left the E site, and now we have dipeptide that formed, and we are ready to keep on reading that message, that messenger RNA, until we run into that stop codon. Okay, so here's your entire story when it comes to describing gene expression. So we have the process of transcription that's happening inside the nucleus, and you can see the processing of mRNA, and then mRNA is translocated into the cytoplasm, and you can see this complex is forming here, and we initiate the synthesis of the polypeptide. And then remember, we have this fun enzyme, amino acid tRNA synthetase, that's going to bind the amino acids to proper tRNAs and deliver them to the synthesis site. And then really quickly, just to compare prokaryotes and eukaryotes as far as transcription and translation goes, notice that in prokaryotes, two processes are happening right there in the cell, because DNA does not wrap around the histones, and there's no chromatin remodeling that need to take place, no RNA processing. So those two processes literally can happen almost simultaneously. As long as you have a small RNA piece already produced, you can actually translate that. In eukaryotes, on the other hand, those processes will always be separated. You have transcription inside the nucleus, RNA processing inside the nucleus, and then translation happens outside the nucleus at the ribosomes. All right, so in the next video, I will walk you through specific steps of gene regulation. We will explore how eukaryotes and prokaryotes regulate gene expression. I will see you then.